Well, good evening, ladies and gentlemen. Welcome to the second in the University of Maryland Center for Environmental Sciences, Chesapeake Biological Labs Science for the Community Lecture Series for Spring 2022. For those of you who are attending for the first time, my name is Tom Miller, and I have the honor and privilege of being the director here at the Chesapeake Biological Lab. We hold these lectures, 5 in the spring and 5 in the autumn each year, to help meet our mission and our mandate from the legislature to explore any aspect of the environment that affects the citizens of Maryland or informs those who make decisions on our behalf. Each season we choose a theme and build a lecture series around that theme. This spring's theme is the urban ocean. Oceans are important to our culture, they're important to our economy, and they are important as sites of intense ecological activity that support the provisioning of food, particularly protein, for large numbers of people in the world. Those interactions are most intense where the ocean meets the land in our urban areas, particularly in the areas of our ports. And so for this season, that is the focus of our lecture series. Last week, Dr. Judy O'Neill gave us an overview of a program called the World Harbor Project, showcasing her work in Baltimore and in New York City. In the weeks that follow, we will look at the distribution of metals, often a legacy contaminant in the urban ocean. We'll then talk about the distribution of oxygen, a vital element to sustain life in the urban ocean. And then in the final week, we will welcome a colleague from Morgan State University, an architect who has spent her career looking at the connection between port cities and the harbors and waterfronts that they rely on. But tonight, we focus perhaps on a consequence of the reason for the existence of ports. Despite our shrinking world as air travel brings us ever closer together, it is undeniably true that the bulk of commerce still occurs by ship. One only had to remember images from last year of the number of container ships waiting to unload on the US West Coast and waiting to load in the ports in Southeast Asia to know the truth of that statement. One of the byproducts of that shipping is the introduction of invasive species. And that's the focus of tonight's lecture. Our speaker tonight is my friend and colleague, Dr. Mario Tamburri. Mario is originally from California, where he got his undergraduate degree, and he's taken a sort of anti-clockwise rotation around the US since then, doing a master's degree at the University of Alabama on the Gulf Coast, and his PhD at the University of South Carolina, where he studied the chemical ecology of oysters. That is, what are the chemical cues that oysters use to decide where they should metamorphose, where they should set. And then from South Carolina, he went back to California, and then we were lucky enough to recruit him for a large project funded jointly by state and federal agencies, which has focused on measurements of environmental quality and solutions to the environmental impacts of shipping. So it is my real pleasure tonight to introduce the speaker, Dr. Mario Tamburri. Mario will come to the screen in a minute. I just want to remind everyone that this is a Zoom conference rather than a Zoom call. As such, we cannot unmute you and invite you to ask your own questions, but we do welcome you to type questions into the chat box, and I will moderate the questions at the end of the session. And as a personal welcome, I'd like to extend my welcome to an old friend of mine, Dr. Sam McClatchy, who is joining us from New Zealand. So good morning, Sam. And with that, I'd like to introduce Dr. Mario Tamburri. Mario?
Well, thank you, Tom. I'll go ahead and that was a really nice introduction. And it's a pleasure to be here to speak with you. Hopefully, in the near future, we'll be doing this in person again. But for now, we'll make do with Zoom. I'll share my screen and should be ready to go. Okay, as Tom mentioned, the theme of this series of lectures is the urban ocean. And so I thought I'd start with this very familiar image, satellite image of the Baltimore area. And it's one of the more obvious examples of urban waterfronts. And in large part, the reason why it looks this way is because of commercial shipping, as Tom mentioned. And so today I'm going to talk about one of the issues associated with international shipping, and that's invasive species. And we've been working hard on trying to find solutions to this problem. So my lecture will look like this. We'll talk a bit about invasive species first, why they're important, what the issues are associated with them, how they get moved around. Then I'll talk about ships and their ballast water and regulations associated with that and some of the solutions we're helping to facilitate. Then I'll move to ship and biofouling, the organisms growing on the side of the ships, and again, talk about regulations that are evolving and solutions we hope to come up with. And then I'll just end with one last slide on some of the bigger picture lessons learned from these sort of activities. So first, invasive species. When you hear about the various environmental issues facing the world today, invasive species always makes the top 3, top 5 list. It's a huge environmental concern. It happens around the world in terrestrial systems on land, as well as aquatic systems, of course, in the water. This figure here is kind of dated. It's from 2008, but it gives you a good feel for how widespread invasive species can be in aquatic environments. These are known harmful or nuisance species, so it's not just all the non-natives, but it's the ones that are obvious problems and how intense or how many there are in different coastal areas. As you can see, just about every coastal environment is impacted. The few areas that are left with white bubbles haven't been studied very closely yet. I think once we start looking more closely, we'll find lots of organisms there. So it's happening everywhere. Sometimes these, or most of the time, these invasions are inadvertent or accidental. In some cases, they are deliberate. Sometimes organisms are brought into a new region for a specific reason, like aquaculture, and then there's an invasion that sort of spreads from there. These organisms can have huge impacts on the ecosystems they inhabit, they move into. They can change the ecosystem structure, its function, and services that it provides. They can cause local extinction of native species in some cases, and typically have a big impact on biodiversity, bringing it way down from where it normally is. In some cases, there are some organisms can have human health impacts or marine wildlife health impacts as well. And then there's an enormous cost associated with them, whether it's prevention or mitigation or control, removal of invasive species becomes a very costly exercise once they're there. Things like crashing of fisheries or interfering with industrial operations because of non-native species. So it's a big deal. Here locally, that we have in the waters of the Chesapeake Bay watershed and Bay proper, over 200 known non-native species that cost us millions of dollars a year annually in management, removal, and mitigation efforts. And I'm just going to give you a few examples, not go through the whole 200 list, but just some obvious examples to give you a feel for who they are here locally. The first I'll start with is one of the oldest invaders probably we have. It's Phragmites. It came over from Europe in the 19th century, and it's this large plant with a feathery plume that you probably see on the waterways here locally, and they typically crowd out native plants and can form these really dense stands that really alter the wetlands habitats and structures and functions. More recently, there's been an animal, a snail that's been introduced. This was first seen in 1998. It's called the Rappa whelk. Came over from Asia. It likes higher salinity water, so it's typically down at the mouth of the Chesapeake Bay in the Norfolk region. And it has high reproductive rates, it lays lots of eggs, and it's also a predator on bivalves. And as we know, we already have depressed oyster industry in the bay, and there are other valuable bivalve fisheries here. Adding a predator to the system is not been a good idea. And another example is the Chinese mitten crab. This was found for the first time in 2005. Hasn't really taken off quite yet, but they are here in the bay. Of course, it came from China. It's a little bit smaller than our native blue crab. It's not good to eat, from what I understand, unfortunately. But some of the problems that associated with this crab is that it likes to burrow into sediments. Oftentimes, it will burrow into riverbanks or levees, causing them to collapse. It also competes for things like bait and crab traps and so on. So it can be quite a nuisance itself. But there's just a feel of some of the players we have here locally, the obvious nuisance species. So how do these different organisms get around the world into a new aquatic system? Here's sort of a fairly comprehensive list of the different vectors or pathways. And they're just in alphabetical order here. I've highlighted and italicized there the couple that I'm talking mostly about today, but there are lots of different ways these species can move around. And I'll just give you, talk about a couple of examples. Aquaculture is one of the bigger ones. It's not uncommon for something like our local oyster to be transported to other areas where people want to start aquaculture operations. Sometimes those organisms themselves can escape and form reproducing populations in the wild. But oftentimes too, there's other organisms associated with them that get spread in the process, things growing on their shells or even things growing inside of them, like parasites, that then cause problems for other species in the new areas they inhabit. Another one that you may hear stories about is the aquarium trade or the pet trade. And there's several examples of these. I'll just talk a bit about the lionfish. You may have heard the stories about how they're taking over in Florida and the Caribbean. They're beautiful fish. They come from Asia. They're very popular for aquariums. They have poisonous spines. And what happens typically is when they grow or maybe grow too large for the aquarium, or people have to move and can't take their pets with them, they don't want to kill the organisms, so they'll just sort of dump them in local waters, not necessarily meaning to cause an invasion, but in fact they do. These are now established and reproducing in Florida and the Caribbean and causing big problems there, because obviously there are a few predators who will take these on, and so there's little control of the growth. The final one I'll just give you an example is what I call invasion by FedEx. This is through the live bait trade. You can go online and find all sorts of interesting baits from around the world for local fishing, and quite literally, with your credit card, have them delivered to your house, 24, 48 hours later. Things like bright red, large polychaetes or fireworms from Southeast Asia, things like that. When they show up, they're typically in these containers with sediments from the environment they were taken from, or kelps from the environments they were taken from, or wet newspaper, or whatever it might be. That medium might be a source of invasions, but what we see happening is fishermen will go out and they'll fish for the day, and there'll be 3 or 4 left in the container at the end of the day, and they'll just sort of dump them over the side, not thinking about what that might mean. So, and there are several other ways, again, on this list here. But if you look at the impact of these different vectors, as far as numbers of invasions go, by far the most important is through commercial shipping. And there's sort of 2 ways on ships that I'll talk about today that organisms can be spread. One is through ship's ballast water, and the other is through ship biofouling. This figure is from a colleague of mine, Greg Ruiz. Many of you may know him. He's up at the Smithsonian Lab, CERC up in Edgewater. He's a collaborator of ours on much of this work. This is just one example, but there's lots of them where people have looked at the different vectors like biofouling on ships or ballast water from ships, aquaculture, and other possible vectors, and in this case, it's through time and the number of invasive species, whether it's clearly from that source or it could be that source in others as well. The story really is that ballast water and fouling are by far the most important, and they're increasing in importance as well. So we need solutions to that. The shipping industry, and Tom touched on this briefly, but it is fundamental to our global economy, to our international trade. It's estimated that over 90% of the world's goods and materials come across on ships. It depends on how you count them, but there's about 70,000 or so ships that are active at any one time. Some are decommissioned, there's new builds. And they're getting bigger and faster through time, and new routes are opening up. Things like the Panama Canal was expanded. Now with global climate change, polar routes are becoming more viable. And so this figure here shows you just an example of how container ships have grown over the years, from when they were first invented in the 50s to present day. You can see that the surface area of these ships has grown quite a bit, more room for fouling. Their ballast water volumes also has grown, so more room for moving organisms. And the faster they go, that means it's more likely that whatever's in the ship or on the ship is likely to survive. So we're just making the problem worse with our new innovations. This map is an image. You can find these online at any one point in time, the different shipping routes and different ships around the world. The different colors are different types of ships. So red, I think in this is like oil tankers coming from Alaska to the West Coast kind of thing. But what this illustrates is that if you look at it this way, of the number of ships and where they are in the world's oceans, really there's no place that's being left untouched by the shipping industry and the potential invaders. So I'll first start with the story of ballast water. I have talked about this in the past, but I'll give you the summary here as well today. So the way it works in prior to steel hull ships, in the old wooden ship sailing days, ballast was typically solid material. In fact, many of the cobblestone streets we have in New England were ballast stones brought over from Europe as ballast on the ships as they came over, and then they released the stones and brought on cargo to go back to the old world. With the newer ships, with steel hull ships, liquid or seawater, ambient water is used as their ballast. And what it's used for is to keep the vessel's draft, it's sitting low in the water, it's trim, maintains stress loads. So depending on where cargo is, you want to balance it out with some ballast water. And it obviously regulates its stability. So when the ship is completely empty, it typically floats quite high and it's unstable. So what this figure kind of shows that the progression as a ship, in this case, a bulker or a tanker, is unloading cargo, it's bringing on ballast water. So it always kind of maintains that trim draft stability. Then once it's empty of cargo and full of ballast water, it moves to a new port and it brings on new cargo and unloads that ballast water. And the problem is, in that water, in these environments, you have lots of planktonic organisms that can't survive these voyages and become invasive. So things like, in this case, copepods and different algae, larvae of invertebrates, for example, are problems lie. So as we started realizing this is a problem, and we started developing regulations around this, we being the broader international and national community, this really took off in the mid-'80s when there were a couple of very obvious and very impactful invasions that I'm sure you've all heard of. Well, at least one of them anyway. Here in the US, the zebra mussel was introduced from Europe to the Great Lakes. It became a very obvious nuisance species. It took off. Huge populations, filtered enormous amounts of water in the Great Lakes. So the water became crystal clear in many areas where it used to be highly productive. It outcompeted fisheries for food. It clogged cooling system of power plants. It's been a real problem. At the same time, about the same time frame, we exported an invader from North America. This is the comb jelly that was transported from Atlantic coast of North America to the Black Sea and the Caspian Sea, and had similar sort of effects. This is a ctenophore comb jelly, and it outcompeted local fisheries. There were really no predators for it. Fishermen would go out and come back with nets full of slime rather than anything was commercially valuable. So these two things brought global awareness to this issue, and regulations started coming out. In some cases before the science, and I'll talk about that as we go forward. But initially, one idea that became popular, and it was sort of an interim approach, was doing mid-ocean ballast water exchange. And the idea is that if you take on ballast water in one port, say you're in Asia, and you take on ballast water halfway through your crossing to North America, or at least 200 miles offshore or something, you release that ballast water you took on in Asia, and you fill up with open ocean water. And then when you get to the next port in North America, you're releasing open ocean water. The idea goes that the organisms you originally collected in a coastal environment are unable to inhabit the open ocean environment. And then the fewer organisms you find in the open ocean are going to, again, be unable to inhabit the coastal environment. It's turned out to be better than nothing. It reduced risk of invasions, but it was far from perfect. Some of those assumptions about the biology didn't always hold, but it also turned out to be quite dangerous. Managing your ballast water at sea can be fraught with problems. This image is ballast being pressed through the decks of the ship, spilled over the side as new water is being brought on. This is the Cougar Ace. It's a car carrier that was coming from Japan to, it was eventually supposed to go to California, I believe. It was doing ballast water exchange and didn't do it quite right. It went over on the side. There are other examples of this. There was a vessel in Savannah a couple of years ago that had a similar sort of problem. Unfortunately, one of the salvage crew that went out there to recover the vessel fell to their death. So there was real emphasis now, like this, we should move away from it. We want to protect the environment. We need a better solution. And so what's come out more recently is that there's regulations that say ships have to treat their ballast water with certified ballast water management systems to meet specific discharge standards of really low risk water being released. And these rules came out several places, but most notably at the IMO, International Maritime Organization, which is part of the UN, and here in the US through the US Coast Guard and EPA. So just a short history on how it happened. About 2004 is when the convention at the IMO was adopted. It didn't come into force though in 2017. There was a lot of details that needed to get worked through, and I'll show you some of those as part of our work. Here in the US, EPA first came out with rules in 2008, was followed by Coast Guard rules in 2012. They both had different jurisdictions. More recently in 2020, the Vessel Incidental Discharge Act, VITA, was turned into law that unified the two different agencies around our regulations. Bottom line is this, to minimize the risk of introduction of invasive species through ship's ballast water. All these groups require that you have a certified ballast water management system on board, something that's been tested and proven to work. And the water coming off the ship has to meet these requirements. What these requirements are is kind of like a clean drinking water sort of approach. You have to have less than known bad things in that water. And when it came to developing this list, this is where the policy making and the science got disconnected. I think it was drafted in a lot of ways because it's easy to write in a legal way, but in some ways doesn't make a lot of sense biologically or ecologically. But what they came up with is that in all cases is that you need very, very few organisms in the water before you release it. Oops, sorry about that. Very few organisms in the ballast water before you release it. So you need to have less than 10 viable or living, and there's a big controversy between those terms, organisms that are greater than 50 microns in size per cubic meter of water. And what that was really targeting is that zooplankton group, those copepods and larvae of invertebrates and the like. and it's per cubic meter of water. So it's a quite a large volume of water, less than 10. During the summer in the Chesapeake Bay, we can find tens of thousands, if not hundreds of thousands per cubic meter of water. So they're saying knock it down 2 or 3 orders of magnitude before you discharge it. Similarly, for what they were targeting is essentially phytoplankton or algae or protists. They said less than 10 living or viable organisms per mil of water, between 50 and 10 microns in size. Again, it's knocking it down 2 or 3, 4 orders of magnitude, depending on where you are. So it's reducing the number of live organisms or viable organisms quite a bit. Problems start coming in is that, there are some zooplankton that are smaller than 50. There are some phytoplankton that are greater than 50. There's lots of phytoplankton less than 10. It's far from perfect biologically, but it's what they came up with. And then they also came up with some rules around microbes or bacteria. One is around Vibrio cholera, because there was some work done, which Greg Ruiz and others were involved with, that found Vibrio cholera in ballast water. And of course, that causes the disease cholera and the human health concerns associated with that. So because that was found in a ballast tank, they thought, let's be safe and say you can't have any Vibrio cholera, or you have to have very little Vibrio cholera coming out in your ballast water. And then they picked 2 other bacteria strains that are really more sort of safe beach swimming, safe clean water sort of indicators, E. coli and Enterococcus. And these levels are pretty similar to what people would use for closing a beach, like I said, for safe swimming. Just so you don't, the idea is not to be releasing polluted water through your ballast as well. So that's where we came in. We got to the game a little bit late. The policies already were starting to evolve, but we wanted to jump in and help find solutions that met those regulations. And we did it under a couple of programs that I run here at CBL. One's called the Alliance for Coastal Technologies that Tom referred to earlier. Another is the Maritime Environmental Resource Center, or MERC. Both these programs are academic sort of groups that are really focused on helping to facilitate the development and adoption of new innovations to solve environmental problems. So we try our best to provide rigorous science to support smart regulations. In the case of ballast water, it might have been a little bit late, but we caught up, I think. We want to help facilitate these new innovations. A lot of the work we do is developing new analytical methods to measure the different things that are put in regulations. And a big part of our work is to act as an independent third-party tester of these technologies. That way they can be approved or certified for use. I'll just give you a few examples of where we had to jump in and start tackling issues in the ballast water world where there really was no knowledge or no science. One was, okay, these numbers are great, less than 10 organisms per volume, whatever it might be. How do you sample a ballast tank to get a representative and a statistically sound sample that tells you with confidence, yes, you're below 10 or above 10? We found early on that just simply doing a grab sample out of a ballast tank is a really bad idea because organisms are not uniformly distributed. They're sort of patchy in different locations in tanks. But you're also looking for very, very few organisms in very large volumes of water. So what we ended up working on and circling around is sampling in line. So as ballast water is being brought on board a ship or discharged from a ship, you sample the pipe that brings the water in or out in a isokinetic, hydrodynamically appropriate way. We also looked at how much water you needed to take to get statistical confidence of those samples. It turns out, especially the bigger organisms, it's a lot of water you have to sample because again, you're looking for very few organisms in very large volumes. We also spent a lot of time on picking through what's alive and what's dead, or what's viable, what's non-viable. It's not as easy to think. Clearly when something is dead, it's obvious, and clearly when something's alive, it's obvious, but there's some in-betweens, like some things, some cells don't move. So we worked a lot on vital stains. Viable, non-viable, we played a lot with grow out assays to see if they're in the right conditions with those organisms actually multiply and grow. Very complicated, a lot of devil in those details. And then we also spent a lot of time, particularly with ACT's expertise on sensors. We worked a lot with compliance monitoring devices. Are there tools out there that could be used to determine if a ship is clearly out of compliance with this ballast water discharge when it comes into a port? So rather than doing all the big sampling and testing that we normally do, is there a quick sensor that can tell you, yes, it's clearly way out of compliance? And so we've been working with a variety of those kind of instruments as well. And these are just a few stories. We have others. Our biggest effort, though, in the ballast water world was to test those ballast water management systems. And I'll describe what those are in a little more detail in a moment, but set up a process for testing those systems in a way that gives you sound data where then the US Coast Guard or other countries around the world can certify those systems for use. And that's where the Chesapeake Bay was a huge advantage. What we ended up doing is building a barge shown here with a couple of mimic ballast tanks on the deck and science sampling vans. And we would place ballast water management systems on the deck of this barge and test it under different conditions. So we would first start in Baltimore, where we have sort of brackish water, and we would run it through trials. And if everything went well there, we would go down to Norfolk, where we can get as close to open ocean conditions as we can without leaving the bay proper, and test it there. And then finally, if it all worked, we would go up to a freshwater system up in Sharptown and test there. What this gave us is not just different salinities, but it gave us different biological communities, different levels of suspended sediments, and all those kind of things. So you can test how well these systems work under a variety of different conditions ships might find at sea. So this is what we call sort of land-based or prototype testing. But what was also required is true shipboard testing. We had to put these systems on real ships and see if they really worked under real world conditions. These are two of the many ships we actually worked on, climbed on board and took samples and did the analysis. You can see the engine room of a ship. This is some of our sample equipment for the larger organisms and so on. We tested these things under a variety of conditions around the world, multiple different treatment systems, and different types of ships as well, with different operational parameters and the like. Don't worry about the table. This is just an example of some of the data we produced for a particular system. This is table 17 out of a really long report that we had to turn into, I think in this case, it was for the US Coast Guard to certify a system. But over the years that we were operating the barge and running these tests, we tested 13 different ballast water management systems from companies around the world, quite literally, a few from the US, but lots from Europe and Asia. These systems included a filter plus UV or filter plus chlorine system, and one that was deoxygenation. And I'll show you again what those systems kind of look like in a second. As part of this, I mentioned the sensors, our compliance monitoring devices. We ended up testing about 9 of those over the years. Most of those were fluorometers. We did some work with ATP or total residual oxygen sensors as well that could be used in that application. So we were hoping that this new issue would bring out really new and creative ideas for water treatment, something that we hadn't thought of before. But the reality is we ended up with standard water treatment for ballast water. A lot of things you would see in wastewater or drinking water sort of treatment. The way these systems kind of work, and here's a couple of examples here, is that they often have a physical separation step, usually filtration. This top one here, these black canisters are filters as a first stage. And then it's followed by oftentimes a chemical treatment, mostly chlorine kind of things like sodium hypochlorite, like bleach, or chlorine dioxide, some ozone. There were some non-oxidizing unique compounds. They didn't really go very far. But in all cases, if you're going to treat with a chemical, a biocide, you also have to neutralize that chemistry before you discharge that ballast water. So you don't want to add a bunch of chlorine to the ballast tank and then release that chlorine into the environment. So you have a neutralizing step as well. There are others though. There's things like cavitation, like you heard about deoxygenation already. By far the most popular out of the physical treatments is UV radiation. So I mentioned they happen in stages, usually have a separation stage like filtration followed by UV or chlorine. Some of these get complex. You can have 5 different phases, but most of them are 2-step. And the water can be treated as it's brought on board the ship in the pipe. It can be treated as it's sitting in the tank, and it can be treated on discharge. The trick really is, from the innovation perspective, is that, if you had unlimited space and unlimited power on a shore sort of facility, this is not that hard of a problem to solve. But you don't. You're working on a ship with really large volumes of ballast water at really high flow rates. Some of the big tankers will pump 10 or 12,000 cubic meters of water per hour. And they'll fill their ballast tanks up in 12 hours. So it's a lot of water moving very, very rapidly. You're also working in port environments that have lots of suspended material. Sometimes they're highly productive, like the Chesapeake Bay. And those conditions change. You could be in a freshwater port one week, and the next port of call is a marine environment. It can be warm, it can be cold. All kinds of different conditions that you have to deal with, where like in drinking water, it's pretty consistent. You're just cleaning it up a little bit, make it safe for use. This is highly variable conditions to work with. And then on the ship themselves, you have limited space. You saw some of those engine rooms, and you have limited power. Plus the ship has vibrations, it's swaying and tilting in heavy seas. So it became a lot more challenging from an engineering perspective. But all that said, through all these efforts, we have now about 43, it might be a little bit more now, certified ballast water management systems here in the US, or US Coast Guard certified, and over 100 through the IMO process from different countries around the world. Okay, now switch gears to the other side of the ship. Early on, the ballast water got all the attention, particularly because things like zebra mussels were thought to be transported as ballast water organisms, whether in larvae form. But the other side of the ship, what's growing on the side is also hugely important, and maybe more important as a vector. I like this picture because it shows a transition from ballast water to these barnacles growing on the side of the same ship. They're both have a possibility of introducing non-native species. You can think about biofouling or fouling organisms on a ship from two different perspectives. And this is a little bit different from the ballast water story. Ballast water was always thought of, and ballast water management has always been thought of as a tax on the shipping industry, at least from their perspective. They get no return on their investment from installing a ballast water management system, aside from complying with the rules. But this is different. For biofouling on ships is a big problem that they also want to address, independent of invasive species. Macrofouling organisms, things like the barnacles that you saw in that last slide, or mussels, or macroalgae, or hydroids and bryozoans and the like, and actually even microfouling or biofilms will increase the roughness of the ship, which increases the drag, and that directly increases fuel consumption. And if you're using more fuel, you're releasing more exhaust emissions. So there's a lot of motivation from the shipping industry to minimize fouling on their own. It also, particularly macrofouling, can interfere with the ship's water systems. Most ships use ambient water for the cooling of their power plants, for their fire systems and so on. So clogging of those pipes is a big issue. This has been a battle for hundreds of years, how to stop fouling on ships. Currently, it's called whole husbandry, and it's really based on anti-fouling coatings. Really, that's the weapon now. And those coatings, the paints that go on the side of ships, are either biocidal, things like copper or zinc-based coatings. Cuprous oxide is one of the more common ones, or they're non-stick fouling release type coatings, things like silicon-based coatings, where if an organism sticks on it, it'll peel off and flow. But it's clear that this approach is far from perfect. There are lots of times where they don't work. There are niche areas on the ships, little nooks and crannies in the rudders and propellers and thrusters, where the coatings are not very effective, especially non-stick ones. If the ship's laid up for a long period of time, fouling seems to grow regardless of the coating you have on board. So there's always been this additional need for in-water cleaning, because what happens is every 5 years you put a new coating on the ship, 5 to 6, 7 years, when the ship goes into dry dock. It's an expensive process, takes a lot of time, but in between things still grow. And so what ships often do is they get cleaned while they're in the water, while they're in port somewhere. Traditionally, that cleaning historically has been a diver with a scraper or spinning brush kind of cleaning carts without any concern about what that does to the environment. If you're just knocking things off the side of the ship in water cleaning, whether it's live organisms or it's the biocides from the coatings, that's not a good thing. But in water cleaning is now being thought of as our solution if we do it the right way, and I'll explain that in a moment. The other side of biofouling is, of course, the biosecurity invasive species side, and there are now regulations catching up. It's about 20 years behind ballast water, but there is some movement in this area. It kind of started in 2011 when the IMO came up with a guideline, a voluntary guideline that just sort of says, hey, here are some tips for minimizing fouling in a way that might stop or reduce invasive species transfer. It was a great first step. It's being revised now, but it's completely voluntary. But more recently, 2017, California came out with its first rules, mostly on reporting. So they know what ships are coming in and how foul they might be, kind of thing. But it's a start in California. They're going to regulate it more strictly in the future. Then in 2018, New Zealand came out with rules that had a bit more teeth. A lot of it was reporting as well, but New Zealand can actually turn ships away. If they judge that you're too heavily fouled, too high of a risk, they can turn ships away. And last year, both Australia and Canada came out with proposed rules or guidelines associated with this. So it's building momentum. What's happening now is what happened with ballast water 20 years ago. So what we want to do is learn from the lessons in ballast water and get out ahead of this issue and provide some good science before the regulations get too far along. So they're made in a way that makes sense and will have a higher chance of success. So between, again, ACT and MERC, our goals are kind of sort of shown here, but basically we're working a lot on the methodologies ahead of time on how we would measure these different parameters so we can inform the rules as they come out. We also going to do the independent testing like we did with ballast water management systems and sensors to help these technologies mature and transition into operation, these in-water cleaning technologies I'll talk about. We want to give vessel owners and operators information so they can make smart choices on their biofouling management decisions. We want to provide the data that's needed for approval or permitting of in-water cleaning if it's done safely and effectively. And again, be the scientific foundation for future regulations. Ultimately, what we're trying to do is to help the industry meet their sustainability goals by, if we can minimize fouling and we can minimize these exhaust emissions, or we can minimize fouling without reducing other contaminants into the environment, then we have a real win-win solution here. So let me tell you a bit about those in-water cleaning systems. These are sort of the two ends of the spectrum of different types of ship in-water cleaning. There's one side that we call reactive in-water cleaning, and that's where you remove existing biofilm, or we call it clean and capture. And these are either diver or ROV cleaning carts that have shrouds around them that captures any of the material they knock off the side, and they suck all that up to debris processing on shore or at the surface. So there already are barnacles, there already are mussels, and you're removing them to keep them clean. On the other end of the spectrum, we have what we call proactive or prevention of microfouling cleaning. And this is, again, either ROV or diver-driven carts, but this is where you do it preventatively. Rather than waiting for the big things to have grown on there, what you do is you periodically wipe off the microfilm or the slime layer. And by doing that, you prohibit the growth of other higher organisms. So you prevent them from ever actually starting. What we're focusing on is how do we know that these things are effective at removing or preventing macrofouling? Are they having any impacts on the environment? If you're trying to capture this material and process it safely, how well does that system work? So those are the sort of questions we're asking and developing protocols around. There are also operational questions beyond the sort of regulatory side of things, and that is, do these cleaning systems really reduce fuel consumption? And also, do they impact the coatings? They're expensive, these paint jobs on the ships every 5 years. we hope that these in-water cleaning systems don't impact their performance or reduce their lifespan. If they're cleaning all the time, maybe the coatings don't work for 5 years anymore. So these are the sort of questions we're asking, and we're spending a lot of time developing approaches to address that. I won't go through all these details. What I'll just say here is that to understand how well these systems perform, these in-water cleaning systems perform, and to understand how to test them, there are a lot of parameters to consider, and there are too many to actually to manipulate and measure directly all the time. There are parameters associated with the vessel, what kind of coating it has, how foul is it already. There's environmental parameters like how clear is the water, how much pollutants are there in the water already. There's parameters associated with the in-water cleaning system. How does it work? Is it brushes that are spinning? Is it water jets? What kind of brushes? How do they mow the lawn? All those sort of things have a big impact on the performance of the system and how we test them. And we can't measure it all. But what we have come up with is a series of protocols now that we're trying to spread around the world as ideas for the best way to do this. They're based off our experience with active work in the past and input from experts around the world. We can't document everything that I showed in the last slide, but we can't manipulate everything we showed in the last slide, but we can document it. So we have the information on the data we produce on its performance is linked back to those different parameters. And for experimental design, one of the fundamental things we decided on is that we really need replication in this. You can't just do this on one ship one time and say that the system works well anytime you use it. So we're saying that you have to test these systems at least on 3 different ships that have some variability that helps us understand how predictive the results are. Again, I won't go into a ton of detail on these, but we're working a lot on how you would actually quantify fouling on a ship before and after a cleaning event and control and treated sites. So we'll put, in this case, divers in the water. We'll measure fouling and control and treated sites before cleaning. The cleaning activity happens. We do it again afterwards, and we quantify both biofilms and macrofouling. We're not going to worry too much about whether they're alive or dead. We're going to assume if it's there, it's bad. We learned our lesson from the ballast water world. But the reality is, if you have a barnacle on the side of the ship, it doesn't matter if it's alive or not. It still causes that drag and roughness and fuel consumption issue. We have different approaches for photographs and point counts. For those clean and capture systems where you're removing stuff that's already grown on there, you can do this in a couple of days, 2-3 days, where you dive the day before, you clean the next day, and the third day you dive again and do the surveys. But for those prevention, those proactive approaches, you really need to follow a ship for 9 to 12 months where you can see potentially growth in the control site and no growth in the treated site. So some of these are going to take some time. When it comes to the environmental safety of this activity, we're spending a lot of time on sampling everywhere, comprehensively, and sampling in particular around the cleaning activity as it's going on. That way we have a comparison of the background conditions of the different parameters versus what's coming off the cleaning unit. And I don't want to spend too much time on all these details, but we are going to look at how well the debris processing and sampling before and after the debris removal step. For things like proactive cleaning, there is no capture step. It's believed that if you're just removing the biofilms, that while there is possibly a risk of invasive species through microbes, it's far less than you would have, or far more important, less important than you would have for macroorganisms. So right now there's no cleaning required, or so no capture required for that cleaning process. The parameters we're looking at from that sampling approach are shown here. We're looking at different biocides of the coatings like copper and zinc. We're looking at total suspended solids, particulate organic carbon and dissolved organic carbon, just to have a feel for how much stuff they're releasing into the environment. Particle size distributions that you can see if it's different than the ambient water. And we're also starting looking at microplastics. It turns out that about 40% of these marine coatings are polymers, plastics. And so perhaps ships are a source of microplastic release in the ocean. We're looking into that in more detail, but maybe in-water cleaning might exacerbate that problem. If there's scrubbing and coatings, there might be a possibility of microplastics building up in these environments. These are just the standard methods that are used for these parameters, which is nice. We don't have to come up with methods. There are accepted approaches for how you measure these different water quality issues. And I mentioned these other operational considerations, impact of coatings. This is a tough one to quantify, but we are able to get things like, scuff marks from cleaning. That's pretty obvious. We're working on an approach now to actually sample the paint all the way down to the steel and then look at leach layers under a microscope to see if they change, but that's still a work in the process. One of the biggest variables of how well this works is the operator. How skilled and well-trained are they? Whether they're a diver cleaning the side of the ship or an ROV pilot has a huge impact on how well it works and how safe it is for the environment. This is a tough one, and we haven't been able to solve it yet on how we quantify operator variability. But the final one, the good news is on fuel efficiencies. We don't have to do this. There are other groups that have already come up with methodologies for that. The ISO, International Standards Organization, has come out with a method for how you look at fuel efficiency on ships. So we can take it off our list. Just quickly, because I know I'm running low on time here, I'll tell you a couple of stories of the two systems we've tested so far, we're testing so far as part of this process. We're building these protocols, but we're also testing systems to get them moving into operations. The first one we did was back in 2018. It's the SGS whale shark system. It's one of those clean and capture. The ship's already fouled, get that stuff off the ship and collect it all. This green hose next to the ship here is the diver with the cleaning unit. This is their processing, the blue container there, and some of our team around sampling and so on. The second one we're working on now, in fact, next week we're meeting the Tisla again in Baltimore. She's a proactive cleaning system where she periodically removes the biofilm layer to prevent the big things from growing. This is our team sampling here at Dundalk. And this is the little ROV going over the side. And I'll show you a little bit more of these in a second. Quick story on SGS. We did a full test of the system to the best we could. The first time anybody ever tried anything like this, and we were able to publish a paper on it and so on. But one of the success stories from this is this data here. I'm not going to go into the details of the data, but basically these are copper levels, either dissolved or particulate copper. And these blue or yellow lines are regulated levels which you are allowed to release into the environment. You don't want any of these bars to be above any of those dotted lines. What we see here is that they were putting out too much copper out of their treatment box where they're collecting all that material. A lot of copper was coming out the other end. They were hoping that wasn't the case because the solution is kind of expensive, but they realize now that they have to treat for biocides on discharge. So what they did is they added a step where they add at the end of the pipe, they added this selective media that binds to metals. This is organoclay. And what it does is as the water goes through, that clay really binds to all the copper, removes it out of the water before it goes back into the environment. So we help them identify a problem. They've now come up with a solution, and hopefully they're much more user-friendly and available for use in different ports now. And I'll just tell you quickly how the whole skater works because it's kind of clever. The company who came up with it is Jotun. Jotun is the biggest paint manufacturer of marine coatings in the world. And they realized their customers need more than coatings. So they worked with a robotics group to come up with this ROV sort of system. And the way this whole system basically works is when the ship goes to dry dock, gets a new Jotun coating on board, they also add this little ROV. And what they do is they monitor different environmental conditions of that ship. And when the conditions are right where they think fouling might start, warm water, temperate environments or whatever it might be, they then deploy the ROV over the side to do a survey. If the ROV sees that there's starting to be more than just a little bit of biofilm, they'll actually go into cleaning mode, and then they mow the lawn and remove the biofilms. They do this while they're in dock. So while they're doing cargo operations, they can lower this over the side and clean the ship. So we're going to be following the ship for another 6 months, and we're measuring fouling and all the environmental parameters to make sure it's safe and effective. All right, last slide. I just wanted to end with some big picture things that we're finding from all this work. One is that the problems that we try to address and their solutions are never as simple as we first think. It's always easy to say, yeah, just count all the dead ones or all the live ones, And devil's always in those details. So while solutions may sometimes seem really obvious, it takes a lot of work to make sure they're successful. And what we found is really critical to the success of some of these solutions is really buy-in from interdisciplinary partnerships between us as scientists, but also engineers, regulators, and the private sector. We all need to work together to solve these problems. And from us as scientists, from our programs in particular, what we found is that transparency and credibility is really critical to the success of implementing these practical solutions to environmental problems. All right, I'll stop there, Tom. I'm sorry I went a little bit long, but I'm still happy to stick around and answer several questions if there are any.
Thank you, Mario. I'm always fascinated by your presentations, and this idea of the Roomba for the cargo ship, I think is really quite remarkable. So let me turn to the questions, and I'll ask the first one from Jack Templeton, and it's focusing particularly on the hull husbandry. And it's, have you had any collaboration with the Navy Lab, the Navy Surface Workflow Center, Carderock Division, West Bethesda? Because Jack knows that they're actively involved in coatings and perhaps cleaning. And as an inside comment, he said he retired from there about 2.5 years ago. So.
Yeah, so we work very closely with them. Bill Hartel and Eric Holm are both heavily involved in a lot of work as advisors. And they have a lot more experience than the private sector does in this. However, Navy ships have different operational needs, and they typically sit a long time and then are very active, and then they go back and sit a long time. So some of the solutions might make much more sense for the Navy than they do for these commercial vessels, which are really always moving, or they should be, to make money. But yes, the Navy's been great at supporting a lot of this work, and they've been learning a lot from the stuff that we've done. They've been taking some of our protocols and adapting into their particular studies.
Just as a personal follow-up, Mario, am I right in thinking that the Navy is exempt from many of these rules and regulations?
Yeah, they are, as are other navies around the world. However, I know for a fact the U.S. Navy, when possible, tries to follow all the environmental rules. So ballast water, they may or may not comply. It is an added expense. The requirements for machinery like that on a Navy ship are pretty hard, pretty significant, redundancies and so on. But for fouling, it's different. Fouling is both fuel consumption and noise, right? So they don't want anything growing on the side of their ships at all, period. And anything they could do to solve that, they've invested in really exotic coatings that include things like silver microparticles and so on that are enormously expensive, but the Navy can afford it. So they're motivated to keep the fouling off.
Yeah, I think as well, in all fairness, whenever I've had any dealings with the Navy, even though they may be exempted from the letter of the law, they choose whenever possible to comply with the spirit of the law. And I just give a shout out to them for that. I want to bring you around to the ports, and you said about these in-water cleaning systems obviously being sort of land-based. I wonder whether you would talk for a minute just about the competition between ports in terms of their environmentalism. I think this is something that always surprises me.
Yeah, it's same with me. We work a lot, of course, with the Maryland Port Administration. I should have mentioned this. A lot of the support for this work you see here comes from MPA, Maryland Port Administration, Port of Baltimore, and the U.S. Maritime Administration. They've been our biggest supporters for a lot of this. And so ports often are competing with each other for cargo operations, and you want to make your port the most attractive as possible. And so in this case, particularly on the in-water cleaning side, when I pitched the idea to the port, they looked at it as a business development sort of solution as much as an environmental solution. They thought if we can stimulate new business development in the port that does this in a safe and effective way, that means more jobs locally, but it also means we could advertise to the different shipping lines that not only can you do effective cargo operations and you're close to the 95 and all that great stuff, but you can also get your ship clean while you're here. And so they've been really interested in this issue, again, both for protecting the waters of the Chesapeake Bay, but also bringing more business here locally.
Yeah. All right. Well, Mario, on behalf of everyone who was on the conference tonight, I want to thank you very much for the presentation. For those who attended, thank you very much for your time. A reminder that we will be back next week at the same time with the third in our 5 presentations. That one will focus on metals in urban oceans. Metals are often a legacy contaminant left over from past industrial practices that still have considerable environmental impact. So I encourage you to join us next Tuesday at 7 o'clock, and we'll see you all then. Thank you very much. And Mario, once again, thanks very much for a great presentation. Good night, everyone.